Yes, I see your slides.
Awesome. So, hi, everyone. We are getting close to the end of the workshop, and we've looked at so many different data types, but there's one that we have not discussed so far, and that's geospatial data. So geospatial data, or geodata, is data that includes information related to locations on the Earth's surface. So you can map objects, events, and other real-world phenomena to specific geographical areas that are identified by latitude and longitude coordinates. And geodata is powerful because it adds dimensions to traditional data, the location, and usually also the timing in terms of day, month, year, etc. So in addition to asking what we always do, which is what data was collected, you can ask where it was collected and when it was collected. And this concept tracks very nicely with classical descriptive epidemiology, where we describe the occurrence of health and disease conditions in terms of three things: person, place, and time. And if you're thinking in terms of molecular epidemiology, where you're looking at samples, say from parasites rather than people, then you can also assess the sample in relation to the place and time in which it was collected. So essentially, adding time and place opens up the possibility of seeing patterns and trends, which deepen our understanding of the world. And that's something we are very interested in, of course, as scientists. A very classic example of this is this person named John Snow, an epidemiologist in London in 1854, where he did some pioneering work where during a deadly cholera outbreak, he started marking cholera cases on a street map, and then found that they clustered around a particular water pump. And this was implicated as the source of a deadly cholera outbreak. So this is a very early and iconic example of using geospatial mapping for epi or biomedical investigations. I did a recent literature search looking for examples that combine geospatial data and genomics and found several interesting papers. So people are using this in many interesting ways. So in this example, snow leopard droppings were collected from a geographic area and fecal DNA extracted to identify individuals. And this data helps to monitor populations of this endangered species. In another paper, blood samples from patients with confirmed Plasmodium falciparum infections were subjected to parasite genotyping and genomic analysis. And they have some very interesting results in terms of parasite migration patterns. And in this last example, samples from tuberculosis patients were subjected to whole genome sequencing and spatial analysis to identify smaller areas with high TB burdens, where you might now put in place targeted interventions to interrupt transmission. Let's get back to ViewPath-DB. And ViewPath-DB has incorporated geospatial tools into a mapping tool called MapVEu on our vector-based platform. So vector-based is a platform that you have worked with in this workshop in the last several days. This is our platform which hosts data related to invertebrate hosts of human pathogens such as mosquitoes and ticks. So this mapping tool is called MapVEu. It has been implemented in this form that I'm showing here on the screen for many years. And it is a powerful way of bringing together three types of data: genomic, phenotypic, and population data. And in its many years of operation, MapVEu has been used in many different ways. So one of them is it's a source of data for meta-analysis. For instance, in this paper from Science, which describes the crash of long-term insect populations, which they dubbed the insect apocalypse, it used data from MapVEu. It's also useful for validating other data tools, such as this mosquito forecasting tool. Data from MapVEu can provide inputs for modeling studies, such as this one. And also it helps to ask this very basic question: who is working in my geographic area? So this year has been very exciting because our developers have been overhauling MapVEu. And 2 months ago, we released an all-new version of MapVEu, which features a user-friendly exploratory data interface. And that's what we want to share with you today. So it is in beta testing, which means you get to try it out and give your feedback, and you can find it from the homepage of Vectorbase. The new MapVEu has many different advantages over the old one, which is that it is very flexible. It has rapid loading, it accommodates new data types and new metadata fields, and also has many different new graph types that you can use to create your own visualizations. And my colleague Sarah Kelly will give you a demo in a couple of minutes, but I wanted to point out some features to look for during the demo. So when you open up the map, you see two main things. You will see a big full screen map with a legend. And then on the left side, you will see this menu, which I've sort of zoomed into here. And this menu is very powerful. It gives you lots of options. So for one thing, you can display data on the map using a variety of different markers, such as donuts and bar plots and bubbles. The second thing you can do is you can browse through all the data in the data sets, see all the variables and the distribution of each of them. And you can filter the data to select a subset that is of interest to you. Further, you can download the data. You can download all of it or your subset of interest, and you can work with it on your own. And you can share your analyses with colleagues. You can write notes on your analyses and save all your analyses in your free Vectorbase account. So we look at how to do all of that. And our work with MapVEu is just getting started. So here are some things that we are working on for the future. We hope to introduce raster layers, which means you can layer additional data such as rainfall or climate on top of genomic or genotypic data. The second thing is you can upload your own geolocation data sets basically that have both geolocation and bioinformatic data, and look at it on your own. And we also want to expand this MapVEu tool to all of the ViewPathDB genomic and other sites. So before we move on to the MapVEu demo and exercise, let me quickly introduce the biology behind the example that we will be using, just so everybody is on the same page. So we are going to be focusing on the parasitic disease malaria, where Anopheles mosquitoes are the vector. And there are two major ways of controlling mosquitoes. One is by sleeping under a bed net, and the other is by spraying insecticides that kill the mosquitoes. And one of the modes of action of insecticides such as pyrethroids is that they affect the voltage-gated sodium channel in mosquitoes. And this disrupts critical cell signaling, and that is what leads to mortality in mosquitoes. But what has happened is prolonged and indiscriminate use of insecticides has resulted in selection of mosquitoes that are resistant to insecticides. And some of the resistance comes from these KDR mutations or knockdown mutations in the sodium channel, where the leucine at position 1014 is replaced by either phenylalanine or serine. And that is why these are the molecular markers of insecticide resistance. And so these two mutations are of great interest to people who study these things. And using our mapping tool, we can ask questions such as, where in the world are these mutations found? Or how is their prevalence changing over time? So let's look at that coming up next. And Sarah, do you want me to run the pre-recorded demo?
Yes, please. Sorry, my internet keeps bumping me off. So I think that's probably the best option so that we definitely get through it.
No problem. So Sarah recorded a demo, which I have here as a video that you can see now on your screen. And Sarah has also recorded audio on this. So just let me know. I'm going to start playing it, but let me know if the audio is... Can you hear it?
No, I'll just speak while I'm here. Okay, so again, this is just showing where to find MapVEu. as Nipur indicated, in the tools menu with the beta tag. And then when the data appears on the screen, the default is all samples in the map. And the samples in the map it's data that we have manually curated. We've gone out and found this data most of the time. It's quite rare for people to deposit data with us. So it's not a full representation of all the species around the world. It is just what the data that we've been able to get our hands on. So that's what you'll see now coming up. Any data deposited in Vectorbase will show up by default. And then on the left, you can see a panel where you can configure your map. So you can show these donuts that you can see now, or you can change it to bar plots, and little plots will come up to show the species count in the plots. And you have the bubble markers, and you can filter and download and share your data. Like Nepal said, and we'll go into more detail of this shortly. Just waiting for my mouse in the video to catch up. And then on the right, you have a legend. At the moment, that's showing species, but it can show anything like your KDR mutations and things. It's like an active legend. And then at the top, that is, again, the default for this timeline is all years of all data in the map. And it's the collection start date for now. If you've got some years that you're specifically interested in, you can highlight it, and that will automatically update the map to show just the points in the data that you're interested in. You can toggle that on and off, and the map will update backwards and forwards to either the selection you've made or the default of all data. Okay, and then just wait for my okay. I think I was just struggling to get it to change at the time. So another thing that I just want to point out is that the legend at the moment it shows the 7 most commonly found species in the data submitted to Vectorbase. So there's a lot of green on that map, which might not seem very useful. And this is something that's going to change. But at the moment, the way to get around this is to zoom into your area of interest, which for us today, this is going to be Africa. So we make that the center of our map. And once that's the center of our map and the map's finished loading, you can go to the panel on the left once it's loaded. Yeah, okay. So now you can see all the points for Africa, and there is some extra area in there, obviously. And at the moment, you can see on the panel on the left that show counts for is that filtered is selected for, which means we have no filters at the moment. So again, that's showing all samples on the map. Whereas if we change it to visible, that's going to just show the counts in this little results table for all the data deposited in the area that's visible on our map screen right now. And so we can scroll through and we can see the most sort of abundant species in our visible area on the map. And we can then go to the filter and we can select for these. So yeah, next we'll go to the filter button, which is on the left. And then you can see that species is a featured variable. So this is going to come up as the variable that you access straight away. And then there's a little search bar at the top there, and you can search for your species of interest. So I'm going to pick all the Gambi complex. So we'll pick those 3. And I'm going to pick Coluzi and Fenestus and Arabiensis as well. And then I'll have 7 species. And you can see as I'm searching for these species, the legend on the right is constantly updating. So you will be able to see your legend update whilst you're doing this. And once they're selected, you can see that there's now at the top end of the little logo, that the mosquito logo, there's a thing that says add filters, and next to it you can now see a grey little bubble that says species in it, and that shows us now that we have started to add a filter for the species that we're interested in. So yeah, shortly that legend will update to show the 7 species of interest. There we go. So you can see them there now. And now when you go back to donuts, this should have a better color representation of the species in Africa. And then you can change that to bar plots. And you'll see these little graphs, which if you zoom in on the map to an area, they automatically update and you get a closer in visualization of the area. And then you can go to bubble plots. And for bubble plots, there is a count. So the size of the bubble is the sample count. So the bigger the bubble, the more samples there are in that area. And then we have this coloration as well. And so I think I'm going to do categorical. Yeah. So for the color comes from proportions for categorical variables and means for continuous. And because Nepal's talked to us about the KDR mutation, that's around where there is insecticide resistance, I'm going to pick that variable now. And we'll be able to, I'm going to star it as well, which means you can favorite it. And once your variable is favorited, you can toggle on and off your favorites. And then later you can find that really easily. So now on the map, you can see, for example, that in Dar es Salaam, it's really faint there. It's a really faint blue color, which means there's low prevalence of the KDR L1014F mutation. And then over in Burkina Faso, that's got a black dot, which means there is a high prevalence of KDR L1014F in that area. So now we're looking at the KDR mutations. We can add this as a filter. So again, I've favorited it, so I've toggled it on and it's now easy to find. So I can select the prevalence of KDR L1014F. And now I want to add a filter for all samples in MapVEu that have a result for a KDR L1014F assay. So I can do that by highlighting the whole of that graph, and that adds a filter at the top. And you can see that my legend is updating. You can see at the top here in this diagram that the little pink bar has changed and it's now smaller, and it's now showing 55, I think, studies out of 700 and something. So it's just those 50 or so studies that have results for KDR L1014F. So now we've added the filter, we can go back to donuts and we can create some plots. So I'm going to go to supporting plots and I'm going to choose a time series. And for you can, make this graph bigger and smaller in the corner. And I'm going to select my x-axis as collection start date. And again, I'll favorite that for later. And then I'm going to choose the KDR L1014F variable. And then I'll overlay species. And you can see that only those species on the right were the species out of my selected species that had results for KDR L1014F. So not all 7, just those species. And you can see there that graph's really clumped up and not particularly useful like that. So there are some really handy tools on the map, which means you can just if you click the auto zoom button on the x-axis range here, then it will automatically do some work for you and make the screen better. And you can see at the top, those are the assay counts. So you can kind of tell how reliable your results might be by how many assays were conducted. So in some of those areas, there are not many, so they're probably not very reliable. And then the lines, they are the prevalence of the KDR L1014F in your species, and the colors are indicated in the legend on the map. But I want to get rid of that point in 2000 because it's kind of ruining my graph. So I can do a custom x-axis range, and I can put in the years that I'm interested in to get rid of things. And that little yellow bar is just telling you where the data has been truncated. It's not an error. It's supposed to be there to show you that you've truncated it. And there's a little message that tells you about that at the bottom. And I might want to, you can change it to week, month, year, and however many weeks or months or years you would like to bin your data by. And obviously you might want to give it a title so you can personalize your plots, and I can save that. And then I might want to download my data. So my genotype, I'm interested in a genotype data. So I'm going to pick in my subset the genotyping assays. And in here, a little pop-up should come up. And I'm not just interested in the KDR L1014F. I want to see if there were the other mutations there, like the S or the C, and if the wild type was there. So I'm going to select all of those so that they are all, I can see all the percentages of mutations in my downloads. So if I click that top one, then you should, on the right-hand side, get a preview of the data that will be in your download. So now I can see that I've got the prevalence of C, F, S, and wild type to download. And you press download, and that's it. You can do some further analysis on your data. If you do want to look at sample data as well, you will have to download the other files and merge them together, but they've all got identifiers. Each sample has an identifier which you can look through each of the files and link them up. So then I might want to share my analysis. So I'm going to name it again. And once you've named it, you can press the submit button. And once you press the submit button, you will be given a link that you can share with your colleagues or your friends quite easily. And then you can also add some notes. You might want to, if you're sharing it with your colleagues, you probably want to give a little bit of an explanation of what you did and the steps you took to create your plot, so that people can always go back to it, and yourself, you can go back to it so you understand what you did. And these will all be saved when you are logged in to your free Vectorbase account in the section called My Analysis, and you can see which one I'm viewing, and you can see all the ones I've made in the past. And that is everything for the new map. But we're happy to answer any questions if you have any.
There was one question in the chat, and they're asking, can I perform such analysis for mutations in host receptors and their effect in disease prevalence or severity in endemic regions in ViewPath?
That's going to be a future. I'm right in saying that would be something for the future. Right now, this is just available in Vectorbase, and it's stuck with it. There's now some mapping functionality in ClinEpi, but it's not made it to all the sister sites yet. But hopefully in the future, it would be something.
Yes, right. So right now you can only use data that's already part of Vectorbase and available to you. But we have another sister site, which we haven't discussed at all in this workshop, called ClinEpiDB, the clinical and epi database. But we have a DIY workspace there where you can upload your own data. It does not have to be clinical and epi data. But if it has geolocation data, then you can look at it there. So I'm happy to send you a link and discuss that more with you. So if you want, just reach out to us and we can talk more about this if you like. But for now, I think, Suzanne, it's 12:56, so I'll probably let you continue.
Okay. Were there any other questions for Nupur or Sarah?
For the specific examples that were mentioned in this presentation, how were the data collected? Were they collected from papers published or?
Yeah, so most of the data with Vectbase is from manual curation of papers. We do have some groups that actively deposit with us. They've started doing it and now they've seen the value in it. But most of the time it is us going to the paper, and then we do try and contact the author to make sure that we have all the information. But yeah, it's more that way. But if you have data, we're always happy for people to deposit it with us.
Great. Well, okay, the KDR mutations is the main mutation, but resistance is also due to metabolic
Yes.
mutations. Do you cover that as well? Or?
Yeah, so that is something that we do cover. At the moment, it's not been a focus of the map, but we do have a lot of that data, and it's all there ready to be loaded in, basically. So the more people we have that are interested in it, the more likely it is to get pushed out. But yeah, we do have that data.
Great, thanks.
Okay. I'm gonna go ahead. Yeah. I'm gonna stop recording.